Welcome to Study Loop.
So here we are looking at epigenetic modifications. So if we first take a look at the term epigenetic, we can break it into two parts, epi and genetic. So epi will typically mean upon, whereas genetic is anything to do with the gene. So these are modifications that happen on the gene, typically before any kind of transcription is ready to occur. So you can imagine you have a strand of DNA with the gene of interest, of course. Now, before this gene is ready to be read, you can imagine that the DNA inside of a cell is very coiled and condensed to allow for the best optimal storage of that genetic information. And when we want to transcribe one of the genes, we have to somewhat unwind that portion containing that gene of interest. So this involves the use of histone proteins, which are what allow it to coil up around proteins that well as the supercoiling involved in the structure. In addition to these, we also have certain proteins that will, say, add certain groups to that DNA strand to either increase or decrease the capability for these to be transcribed. And this process is often called the methylation of DNA. So in some circumstances, it will inhibit proteins from binding because these methyl groups will block it. And in other rare circumstances, it can actually enhance the reading of the gene. And these are what we call those epigenetic modifications, because it is before the gene is read and it is on and upon modifications of the DNA strand itself. So here, when we look at the answer choices, the best answer choice is going to be A, because these are going to be modifications to the proteins allowing for the supercoiling, which are the histone proteins, and for other modifications such as, say, the methylation of those DNA strands. So here you should pick answer choice A.